Namaskaram. Welcome to Conceptual Life Science. This channel is dealing with clearing of the concepts in the syllabus of CSIR. So we've started with the third unit, that is fundamental processes, and in the third unit we've started with the replication topic. Now, or based on this topic, this is a third lecture on prokaryotic DNA replication. Today what we are going to see is that the role of FtsZ in septum formation. What is FTS? FTS stands for temperature sensitive filamentation. Okay, if under certain condition, non-permissive conditions of temperature, the bacteria can undergo mutations and you have a formation of mutants called as FTS mutants, okay, wherein the bacteria forms long filaments. So there is a mutation in the cell division cycle or in the division apparatus and you get to see filaments of bacteria. There is no septum formation. So in today's lecture, we are going to see the role of FTS protein in septum formation. Actually, it's universal that the bacteria should undergo septum formation before it goes cell division. So if you see almost all bacteria, especially the rod-shaped bacterium, they do undergo septum formation, and unless and until septum formation takes place, the bacteria cannot move on for cell division. So in today's lecture, we are going to see how the septum is formed, and this septum is in the form of a ring called as Z ring. The proteins which are involved in formation of septum. Now, you know that proteins are coded by genes. So, you have a couple of genes which will code for proteins which are involved in septum formation. As I said that septum is utmost important. The cell has to undergo septum formation and then there is the actual cell division taking place. So, which are the genes or which are the proteins involved in septum? In case of bacteria. In case of prokaryotes, there are ample number of proteins. Actually, it's a multi-protein complex which are involved in formation of septum. You don't have a single gene or a single protein which is involved in septum. You have a couple of proteins or rather a multiple protein complex is involved in formation of septum. So that all we are going to see one by one today. So let us start. The gene which is involved for septum formation or the is FtsZ. Now here small f stands for the gene and capital F stands for the protein it is expressing. So small FtsZ is the gene involved in expressing a protein called as FTS, capital FTS. So the product of FtsZ is capital FTS, you can say. Now this protein is required for septum formation. So initially this protein is very important, of course FtsZ, but there are a couple of proteins like ZipA protein. You have ZipA protein, you have FtsA protein, you also have a protein called as FtsW, you have FtsI. Okay, so you might find a bit confusing, but as you move on, you will understand that they are all very simple. So you just have four proteins forming septum or forming a Z ring. Okay, so FtsA, FtsW, ZipA, and FtsI. So these are the four main proteins involved in septum formation. But out of these also the key role is played by FtsZ. Now this FtsZ protein is actually having a cleavage activity. It is a GTPase. Okay. So this FtsZ protein will cleave a GTP molecule and the energy liberated from this GTP molecule will be utilized for the polymerization of FtsZ. So this is the key protein which is involved in forming a ring shape. Okay, so FtsZ protein is somewhat similar like the tubulin protein in eukaryotes. FtsZ resembles the eukaryotic tubulin. Okay, I mean its function is almost similar to the tubulin protein in eukaryotes. So FtsZ undergoes polymerization and it forms a ring inside the bacterial envelope. So bacterial envelope consists of cell wall, you have cell membrane, and in some capsulated organisms you have capsule also. but basically you have cell wall and cell membrane. So this septum formation or this Z ring which is formed due to FtsZ protein, it organizes itself in the mid cell. Usually the septum formation occurs from the center of the cell. You will hardly get to see that the septum occurs unequally. Okay, that all we have covered in the previous lecture also. So you can check the previous video that why the septum formation takes place exactly in the center. Okay, so this Z ring is formed in the center of the cell, and Z ring is nothing but the septum. Okay, so this septum is formed in the middle of the cell and it arranges itself below the bacterial envelope, that is below the cell membrane. So you have a couple of enzymes which are involved in septum formation. So their main role is cell division. So after septum formation, cell division will take place. Now in the earlier lecture also we saw that if there is mutation in these proteins which are involved in the shape of maintaining the shape of the bacteria, then you will have mutants, okay, which will have various shapes, like you will have mini cells, you will have filamentous cells. Okay, similarly here also, if there is mutation in the gene involved in forming FtsZ protein, then obviously there won't be septum formation, because the key role of FtsZ protein is to form septum. Okay, so if there is mutation in this FtsZ gene, then obviously it will block the septum formation, and if there is no septum formation, obviously you will have cell division, but the cells will be having a long filament like structure because there is no septum. But if the FtsZ gene is expressed maximally, or if it is overexpressed, then what will happen? Then it will lead to formation of septum. Okay, if FtsZ is produced in large amount, then there will be large amount of septum formation occurring, and the cell will undergo frequent cell division. Because of that, you'll get small mini cells formed. Okay, so these are the two opposite things we'll get to see, that if there is no expression or if there is mutation in FtsZ, then it will lead to formation of filaments. But if there is overexpression of FtsZ gene, then you will have lot of septation and it will lead to frequent cell division leading to mini cells. Okay, so these are the two things we'll get to see in case of mutations in FtsZ. FtsZ, as I said, is not the only protein involved in septum formation. Along with FtsZ, you have a couple of proteins called as ZipA proteins. Then you have FtsA protein, you have FtsW protein, and you have FtsI. So somewhere you can jot down this, the key proteins involved in septum formation. FtsZ, of course. Following FtsZ, you have ZipA, then you have FtsA, you have FtsW, and you have FtsI. Okay, so these proteins are also involved in septum formation, or these are the units, you can say. This together will form a machinery for septum formation called as divisome. In short, you can say that these are cell division proteins. So these are the proteins responsible for synthesis of a new septum. Now, where is this FtsZ located? If you consider the bacterial cell, this protein usually lies in the cytosol or in the cytoplasm. So the dots here which I have shown in red, blue, green and black color, these are nothing but these are molecules of FtsZ protein. But otherwise, before cell division or when the cell division is not occurring, these proteins lie in the cytoplasm or in the matrix or in the cytosol of the bacterium. But when the cell division is going to occur, these proteins, they rearrange themselves in the form of a ring. And this ring is called as Z ring. Why? Because the main protein in this ring is Z protein, that is FtsZ protein. That is why this ring is called as Z ring. Okay, before cell division they arrange themselves. How? They move circumferentially. Okay, throughout the circumference they move in the mid cell position. So this is the mid cell position, this is the middle of the cell. Okay, that is mid cell position. So in the mid cell position, these FTS proteins, they arrange themselves around the circumference of the cell but internally to the cell envelope. That is from the inside of the cell envelope, even inside of the cell membrane. You have cell wall outside, you have cell membrane, and you have cytoplasm. So in the cytoplasm, they'll form a circumference. In a circular manner, they will arrange themselves. So you must have understood now by now that FtsZ ring is not a entire what do you say, it is not an entire circle. You have proteins by proteins, or you have molecules of FtsZ proteins arranging themselves in a circular manner. Okay, so it is a discontinuous ring, you can say. It is appearing as continuous when you see under conventional fluorescence microscopy. The ring actually appears continuous, but it's not continuous, it is discontinuous because the clusters of FtsZ proteins. So the assembly defines the position of septum. Now, if you see in a typical cell division cycle, when is the Z ring formed? Like how you get to see the chromosomes in eukaryotic chromosomes only during cell division or only during mitosis. It's just like that. As I said that these FtsZ molecules, they lie in the cytoplasm, but they arrange themselves in a circular manner during cell division. So we can see this Z ring just uh, 5 minutes, 1 to 5 minutes after cell division. Now you'll say that after cell division, why the Z ring is formed? Actually, the Z ring or the septum formation should occur before cell division. Yes, the septum formation or the Z ring does occur obviously before the cell division, but I'm talking about the previous cell division. Okay. So see, for example, you have a bacterial cell, and this cell is now going to undergo cell division and form two daughter cells, right? So when this one cell divides into two, just before this cell divides, how much time? 5 minutes before. Say 5 minutes before this cell divides, it is going to form a Z ring in itself. Okay. So this is the Z ring formation or the septum formation in the parental cell. Now, after the parental cell divides 5 minutes later, then in the daughter cells, this Z ring formation will take 15 minutes later. So you have these two daughter cells formed from the parental cell. Now in the parental cell, just before cell division, 5 minutes before it will start. Now in the daughter cell, obviously, this daughter cell is now going to divide after 20 minutes. You know that the doubling time of bacterium, generally the doubling time is 20 minutes. That is, the time required for a cell to double itself is 20 minutes. So these two daughter cells are now further going to divide 20 minutes later. So 20 minus 5. 20 minus 5 is how much? 15 minutes. So as I said that 5 minutes before the cell division, cell undergoes division, septal formation takes place or the Z ring is formed. Similarly, in the daughter cells also, as the daughter cells emerge from the parental cell, 15 minutes later, that means 5 minutes previous to cell division, you will get to see this Z ring. That means very simple. After 15 minutes, this septum formation will take place. Z ring will be formed, and once the Z ring is formed, this Z ring will get adhered to the plasma membrane or the cell membrane, and it will constrict this cell membrane. Okay, like this. So once the Z ring attaches to the cell membrane, it will pinch off. Okay, it will constrict the cell wall and the cell membrane of the bacterium, and the two daughter cells will be formed. So 15 minutes later, this septum will pinch the bacterium, okay, the cell wall and the cell membrane of the bacterium, and the two daughter cells will separate. Now, as I said that this FtsZ protein resembles the microtubules in eukaryotes. Okay, so exactly the role of FtsZ protein is just like that of microtubules in eukaryotes. So it undergoes polymerization. So here in center I have shown this Z ring. Now this Z ring appears to be continuous, but it's not, as I said so, because the Z ring molecules, FtsZ molecules, one by one, they will assemble themselves in this circular manner. Okay. So you have FtsZ monomers polymerizing themselves in the ring. Okay. So oligomerization or polymerization of FtsZ protein will occur with the help of energy derived from cleavage of GTP. Okay. So you have guanosine triphosphate. So GTP hydrolysis will take place over here, and the energy released from this GTP hydrolysis or GTP cleavage will be utilized by these FtsZ molecules to polymerize themselves. Okay. So this is very simple because FtsZ, as I said, has got GTPase activity. It's an enzyme. Okay. Now, along with Z ring, you have other proteins also. As I mentioned earlier, you have ZipA and FtsA. So here in the diagram, you can see this ZipA protein, and you have FtsA protein also participating in the ring formation. Now, if you see, the key protein is of course FtsZ, but ZipA and FtsA are also equally important. Why? Because if both of these proteins are absent in the bacterium, then the FtsZ protein cannot function, or the septum formation will not take place. So either one of these has to be present. Okay, it's mandatory that ZipA should be present or FtsA should be present, any one of these. But if both of these are absent, then FtsZ cannot form a septum on its own. Okay, so there will be no septum formation, and it will lead to the formation of filaments. Okay, because if the septums do not form, see, this is a cell and you have a septum, right? So after septum formation, the cell divides into two. But if there is no septum formation, then you will have a long cell. Okay, you will have a filamentous cell formed. So now, in case of FtsA and ZipA and FtsA, here I have shown both of these proteins near the cell membrane. Okay, inner membrane is the cell membrane, outer membrane is the cell wall. Okay, so actually ZipA is an integral membrane protein. Integral membrane protein in the sense is that its location is in the membrane. Okay, how you have the bacterial enzymes located in the cell membrane, same way here ZipA is an integral membrane protein, but FtsA is a cytosolic protein, meaning its location actually is in the cytoplasm, but most of the time it is found associated with the membrane. That is why I have shown ZipA and FtsA over here. Okay, so this you must understand. Now, why is ZipA and FtsA required? As I said earlier that both of these proteins are essential for the formation of Z ring. The reason is very simple. Here I have shown the polymers of FtsZ. Now, ZipA adheres the FtsZ protein, and FtsA also adheres the FtsZ protein to the membrane. Okay, so in a way these two proteins are helping the FtsZ molecule to get adhered to the membrane. Now why has this FtsZ molecules to adhere to the membrane? Why do they have to adhere to the membrane? The reason is very simple, that if you have the Z ring starting forming from the center, it should extend. Okay, the septum should extend to the membrane. Unless and until the septum or the Z ring reaches the membrane, it is not going to invaginate the cell wall and cell membrane. Okay, why this ring formation should take place? Okay, so shape and cell division or septum formation will go hand in hand. So FtsW is a member of SEDS family. SEDS stands for shape, elongation, division and sporulation. So it's a group of protein again involved in maintaining the cell shape. So please check out that. Now FtsW, though it is a member of ZZ family and though it is playing a role in cell shape, here it is playing a role in septum formation. Okay, that you must understand. Now FtsW is coded by a gene called as small FtsW. I said in the beginning that small f stands for the gene and capital F stands for the protein it expresses. Now this small FtsW gene is a part of again an operon. Okay, as I said, so now filaments of FtsZ protein will anchor itself to the membrane. Okay, so this ring formation is the first step in cytokinesis. Unless and until ring formation takes place, septum formation will not take place. Why? Because this ring will extend itself in both the directions. Okay, here I have shown the axis, cell axis direction, right, in the cell membrane as well as in the cytoplasm. So the ring formation will first start in the cytoplasm, but slowly step by step it will extend itself towards the membrane and towards the cell wall, because it has to touch, right. The lower end of the ring will get attached to the lower side of the cell, or the south pole you can say, and the upper end of the ring will adhere or anchor itself to the north pole of the cell, right. So that is how the ring slowly it will extend in both the directions. Oh, okay, so its axis will be in both the directions. Simultaneously you will have this ring formation. Some proteins will move towards the north pole and some proteins will move towards the south pole. So the ring is actually not continuous. Okay, it appears to be continuous, but actually you have these clusters of FtsZ proteins and other proteins. Okay, slowly they will find entry. Okay, they will join in the ring formation. So this you must understand how the ring formation takes place and how the organization of the Z ring occurs in the cytoplasm. Z ring form, it's difficult to study in vitro. Now this is always happening in vivo in the bacterium, but how this has been discovered? Okay, you'll have to first study that in vitro, that how do these proteins, FtsZ proteins, polymerize themselves. The problem in it is that the architecture of this FtsZ protein is difficult to study, and that is why it's very difficult to study the Z ring structure. Okay, because its architecture is not understood very well in vitro. In vitro, what happens is that these proteins, instead of forming a ring, they form bundles, right? So they form bundles, and so we are not able to see how it must be, how the process actually must be happening in vivo. So in E. coli this was studied, okay, and in E. coli it was found that this cluster of FtsZ proteins, they arrange themselves, okay, and the average length of this is 120 nanometer and the width of it is 100 nanometers. Okay, so the clusters move themselves, okay, circumferentially. You have these FtsZ filaments formed, okay, and now these filaments will form a patch or it will form a cluster. So you will have clusters of FtsZ filaments, okay, and they will arrange themselves circumferentially inside the cell envelope. Okay, now though these clusters are formed, okay, in patches, this FTS filaments are formed and they are arranged in circular manner, you need to connect them, right? Suppose if I have this cluster, right, this clusters of FTSZ protein, okay, now each cluster will have filaments of FTSZ. Of course, others are also there, but the key protein is FtsZ. So I have these filaments of FtsZ forming one cluster. Like that, you have several clusters, okay, arranging themselves circumferentially. But you need to cross-link these filaments. These FTS filaments need to be cross-linked. Otherwise, what will happen? Some will move outside the circumference, some will move towards the radius, and it won't be a typical ring formation, right? So these clusters are ringed, okay? This cross linkage is responsible for the regulation of these clusters, I can say. So you have a cooperative assembly of FtsZ. Now who all cooperates? You have FtsA, you have ZipA, you have FtsW, and you have FtsI. These all four proteins cooperate FtsZ in forming the Z ring. Okay, so it's a cooperative assembly. No single protein is involved. You have couple of proteins playing a role. That you must understand here. So in case of E. coli, what you get to see is that these clusters, they move at a velocity of 30 per second. Okay, so in E. coli, each cluster contains, okay, these clusters which I have shown here, clusters of FtsZ molecules. These clusters are of about 683 plus or minus 439. That is, you have approximately 240 molecules ranging from 240 to 1100 molecules of FtsZ arranging themselves into clusters. Okay, and these clusters move in both the directions. As I said that you have this axis. Okay, so along the axis, okay, along the central axis, these molecules move in clusters, and these clusters move circumferentially inside the cell envelope. Okay, and the rate of their movement, or the velocity with which these clusters move, is approximately 30 per second. So these clusters, they remain for about 15 seconds before they disassemble and again reassemble. Okay, so a new cluster is formed, but before the new cluster reappears, these earlier clusters remain for about 15 seconds, and then they get disassembled and they form a new cluster. Okay, so you find that this FTS filaments, they are very dynamic and they move at a rate of 30 per second, and I think which is quite high. Approximately uh, 8 amino acids which will link the N and C terminal. So these amino acids in the center are called as linker amino acids because they link the N and the C terminal. So usually the length of the linker amino acid is about 8 to 10, but in some uh, bacterium like say E. coli, this linker amino acids range from 50 amino acids. So these linker amino acids will link the N terminal and the CCT-P terminal. So this is about the architecture of the FtsZ protein, and each domain, like you have the N domain and you have the C terminal domain, or the in between domains are also very important, or you can say they are very critical in cell division. FtsZ filaments, as I said, they get attached to the cytoplasmic membrane. Okay, so you have these both ends of the ring, the lower end attaching or anchoring to the lower membrane, and the upper end attaching to the north pole of the cell. Okay, so this is how they arrange themselves, and the septal ring gets anchored to the cell envelope, and then it pinches off the cell wall and the cell membrane lying beneath it. Okay, so there will be invagination of cell wall and cell membrane inside from both the sides. So you have the Z ring in the form of patches. Okay, there are multiple patches of this FtsZ filaments. Now these filaments are joined to each other. You have these patches, or you have these clusters of FtsZ filaments, but they need to be joined. Okay, so this joining or this cross linking is done by another couple of proteins called as ZAP proteins, ZAP. Now Z stands for FtsZ, A stands for associated, and P stands for protein. So you have ZAP proteins which are always associated with this FtsZ proteins, and they aid in cross-linking these clusters or these patches of FtsZ filaments. So unless and until these patches or these clusters or these FtsZ filaments are cross-linked, they will not form a concrete Z ring. Okay, so for formation of Z ring, the ZAP proteins play a very important role. So if there is deletion of this ZAP proteins, then there won't be any cross-linking between the lateral FtsZ filaments, and since there is no linking, there won't be any Z ring formation, and since there is no Z ring formation, there won't be septum formation, and subsequent cell division will be affected. So you will see that in this entire talk of today's, you have every protein playing a key role in forming the Z ring as well as maintaining the Z ring and the septum. Okay, so septum formation will take place with the help of the ZAP proteins as well as with this FtsA, FtsW, ZipA and FtsI protein. Okay, so all these couple of proteins, or you can say this multi-protein complex, is involved in formation of Z ring and in maintaining the septum formation also. Okay, so today's talk is also very important, and you will have to correlate and link with the previous lectures also. The upcoming lectures are also linked with this lectures, okay, with today's lecture. So please don't miss any of these lectures. Check the previous videos also, because they are dealing with the shape of the bacterium. Today's lecture was dealing with the septum, how the septum formation takes place and how the ring is formed. And in the upcoming video, we are going to see the genes or the proteins which will regulate the location of the septum. Why the septum is located exactly in the center, in the mid cell, okay? Why not towards right or left? So the genes involved in that we are going to see in the upcoming lecture. So I hope you have understood today's talk, and if you have understood, please share this video with your friends also. Let them also get benefited. So my aim or purpose of this lectures is that you clear up all your concepts of CSIR net syllabus. Okay, once you're conceptually very clear, then that will help you in solving the problems. Okay, so that's all for today. Thank you.